abdominal B is expressed mainly towards the posterior. Abdominal A expression is extended out a little bit further into up to the A2 abdominal segment. And ultrabithorax is expressed all the way up to the thoracic segment. So you have these three transcription factors expressed at different concentrations and in different spatial locations. And they're all important in specifying which segment is what. Okay, so in the normal wild-type embryo, you have all of your distinct segments present. But if you're missing the bithorax complex genes, what happens is you get an embryo that basically has a bunch of repeated thoracic 2 segments throughout the body, from anterior to posterior. Okay, so the absence of all of the bithorax complex genes results in repetition of this T2 segment. This embryo doesn't live very long, but this is what you observe. Okay, so if we add back bithorax complex genes one at a time, we can kind of see how they each function. Okay, so if you have a embryo that lacked all of the bithorax genes, and then you express ultrabithorax only, you get now the T2 and T3 and the abdominal segments starting to be developed, but then all of the posterior up through A9 basically have repeated abdominal 1 segments. Now if you take this embryo and you add back the abdominal B, you can start to see, okay, well, abdominal A, and you look at these sections, now we get all the segments we expect up to A4, and then from A4 back, you have a repetition. And then, if you have another embryo where you're lacking ultrabithorax, but you have abdominal B and abdominal A, you get all of the posterior segments, right? A2, or you're missing A1, A2 through A7 and 8. But here is where you see the replication of the thoracic 2 segment, and this grows up to be a fly that has a double set of wings.
So for this diagram, they took all of those genes away and then added them back one by one?
Yeah. So you could tell them add back, or you could try to knock out you could knock out these 2 genes. So it's probably done both ways. So loss of function, gain of function. But basically, so you can see by looking at the phenotypes of the mutants that are missing, 2 or 1 of these complex of genes, which abdominal sections rely on those specific homeotic genes for their specification. Okay, so easy. We're early today, even though I added all of this stuff. Okay, so for the antennapedia genes, similar experiments were done. And I couldn't find the pictures, so I'm just going to back up because we're early. Okay, so this antennapedia mutant, basically what has happened is they took an embryo, and antennapedia is normally expressed in the thorax region in these segments. So they took the antennapedia gene and they genetically engineered the embryo so that it would express in the labial portions. And instead of the antenna, they got legs in the head. They did the converse experiment where they took this labial gene and put it in the thoracic region, and instead of legs in that segment, the embryo had antenna coming out where its legs would be. So it's not as drastic a phenotype as the bithorax complex mutant where the whole segment gets replicated. But there is definitely something that is defining position that is controlled by these genes. And it's also controlling the fate of those cells. So instead of a leg, it's thinking that it has to be an antenna even though it's in the leg region. Okay, and so this is kind of the interpretation of space and time is one of the kind of the mysteries of developmental biology. Like, why doesn't this labial section just make the whole thoracic section become like the mouth parts? I don't know. But, and why does it decide to only affect the leg? You know, is it because the antenna is an extension and the leg is an extension? I'm not sure. Okay, so in the end, all of these segment genes, selector genes, and their patterns are laid down from the beginning, from the maternal genes in the embryo. Okay, so if you don't get the right pattern, early on you're going to have embryos that are already changed, but you can also get to a certain point in development and then have an alteration or a misexpression of the next gene in the hierarchy and then get a strange phenotype as well. Okay, so to summarize where we've gone so far, gap and parallel gene expression controls the expression of parasegments. The selector genes in the homeotic gene complex are expressed along the anterior-posterior axis, and there's two sets, antennapedia and bithorax. And so these complexes are important for specifying identity in the head and the thoracic segments, and these are important for specifying identity in the second and third thoracic segments and the abdominal regions. Okay, so that's all of the material for the exam. So what we could do is, it's 3:40, we could take a break and go through these review questions, or we can wait until I can let you go, and then we can go over them next week. I'll leave it up to you. We can do it now and do it on Tuesday? Yeah? Okay, so let's take a break. Let's take a 5-minute break, and then we'll start.
I had a question. You know how what was it, the homeotic genes? Are they like mutant genes?
Are they mutant genes?
Oh, I could just.
If they are misexpressed, then